student, Assalamu alaikum. This is Laiba Tariq, your biology instructor from tutoria.pk. Today we are doing your chapter 23, that is the biotechnology. Let's start with the fill in the blanks. So the first fill in the blank is the use of polymer chain reaction create a dash of copies in a laboratory test tube. Million of copies. Second है dash free living organisms in the environment that had a foreign gene inserted to them. तो इसमें आंसर है transgenic. Third है dash noun sequence of DNA that are used to find complementary DNA strands can be used diagnostically to determine the presence of a particular gene. Probe. Fourth है dash is the production of many identical copies of a gene. Cloning. Dash are the self-duplicating rings of accessory DNA in the cytoplasm of the bacteria. तो इसका आंसर है plasmid. इसके साथ ही हमारी fill in the blanks complete हो गए. In the next video we will discuss the MCQs.